Okay, good morning. Hi, 101. Week 5. I'm thinking this might be the best week that we have. It's a little nerve-wracking when you have Yeti next to you. Brown Yeti is still asleep, so we couldn't rouse Brown Yeti. He's in the cave. All right, week 5, lab. What you should have done already, but it's okay. You just want to do it right now before you even start. But you want to have a hay infusion. What is a hay infusion? That's the first part of this lab. You need to get like a jar, like a pasta jar, and hopefully you don't have a white Yeti that's messing with you. Oh God, he just squirted that. So don't do that. So don't follow anything that this Yeti's doing. So you're gonna go outside. We made a little video. Hopefully you saw Yetis make a video, but the big thing is you put water in your jar, but the water needs to be unchlorinated. That means that you need to leave the water out. If you use your tap water, leave it out for 24 hours uncovered and the chlorine will dissipate. Otherwise, you can use like water from your fish tank. But anyway, that water, it's important that it's dechlorinated. And then just grab a whole bunch of leaves from outside. You're gonna make, at the bottom of page 2, you're gonna record what you put in your hay infusion, and oh boy, you're gonna record it and the size of the container, like are you using a jar or that kind of thing. Oh boy, and you don't wanna drink it. You don't wanna drink this. Don't drink the hay infusion. Don't drink the hay infusion. Okay, and then you're gonna take a picture of this, the hay infusion. So that you're taking several pictures. You're gonna take a picture of this hay infusion when you make it, and you'll put that in your lab, okay? Day 1, so you want this to sit for like 2 days. So day 1, during the week of lab 5, let this sit for a couple days. It's gonna smell. I would put it outside. Do not cover it. There's little organisms in here. Yours is gonna smell. Okay, so you need your pocket microscope, and this is going to be, after it's sat for just a couple days, you're going to use the depression slide that you got. You're going to make a wet mount. What this is you're going to grab, I like to go way down deep where there's a lot of organisms down in there. You're going to grab a drop, put it in the slide like that, right? You good with this, Yeti? Oh gosh, no. And then you actually can use your, your pocket microscope. I've got a couple different pocket microscopes. Oh boy. Oh, you want to put some on there? Okay. Yeti doesn't use a pipetter. You can use, this is the one that you got from the bookstore, and this is the one, if like you bought it from Amazon. So you're going to use this, put it on the lowest, move it to the lowest, like 150x, and then you move this thing back, and you're actually going to put it right on top and get it into focus. And remember, this is the focus. And what you want to try to do, I'm not even going to show Yeti, is try to get it over to the 250x in focus. Okay, so I'm actually going to set it on top. Am I getting wet here? Oh gosh. Oh, with the hay infusion? Oh, sick. You are sick. It's a sickness. Okay. Once I have it in focus, oh, I see some organisms. If you want to identify your organisms, you can. That's not part of the lab, but on the first page there's a link to the Ward's ID of protists, which are there's a lot of protists in here, single-celled eukaryotes. But you don't have to identify it. What you want to do is just find some good-looking organisms here. You might see flagella. They're moving around. You might see nematodes, which are worms that are moving around in your. You're going to take a picture of this with your camera. You're using my camera to film, but you can put your camera right over the top. You'll take a picture of this and just hang on. Now we're done. What are we going to do? What else are we going to do? You can also, so you're going to make some observations like what did you notice, that kind of thing. You can use some of the slides that are in here too if you want. You don't have to use the depression slide. Okay, so you've taken a photo. This photo that you just took is called Hay Day 1. Okay, so just remember that. Just hang on to your photo. Now you're going to let this sit for 5 days. Okay, so you need the full week for this lab. You're going to let this sit for 5 days. This is going to smell. Oh gosh, what is White Yeti doing over here? We don't know. after the 5 days, you're going to, in your stinky hay infusion, there's going to be a lot more life. Oh boy. And you're going to make another wet mount. You can see here I just put some drops from my hay infusion on here. If you want to put a cover slip on top, you were sent cover slips in your, but you don't have to. Like, let's say you lose everything from the kit, you actually can just take a spoon and put a couple drops into a clear plate and you're fine. This is just fancy. Okay, so if you didn't get the lab kit, it's okay. I'm gonna do the same thing where I look under the microscope at the slide. I'm gonna do the same thing, use my pocket microscope. This time, I can actually slip the slide inside here and start looking around. Always start with your magnification on 150x because it's just easier to see, right? Because you're not as magnified. You're gonna take another picture of this. Actually, it says 2 samples. You're gonna take a picture of this, and then you're actually gonna make another sample. So you make a second sample. Look at me what I'm doing, not Yeti, not White Yeti. White Yeti is not the brightest. You'll take another sample, and you're gonna actually take 2 pictures, okay? This is called, one of these 2 pictures, you're gonna call it Heyday 5, okay? Now, you're gonna share these pictures with your classmates. So you're gonna put them in your lab, but the other thing that you're gonna do, and ask your instructor if you're not in Hess's class, but ask your instructor, let me show you on my computer. In the lab itself, there's a Google Doc link. And so when you're at lab week 5, there's a Google Doc link that you'll go to, and it will take you, it says upload your pictures. You should have 2 pictures, one from Heyday 1 and one from Heyday 5 that you're gonna upload. And just remember to put your name, so you're gonna put it right into here. You'll just upload these pictures right into here and put your name. That way, all of your classmates, we can do a little activity together where we can talk about what you found, and who knows, you know, maybe I can identify some of them. Oh gosh, don't let your Yeti drink, or don't let your Yeti near the hay infusion. No, Yeti, no. Maybe later. Okay, so once you're done with this, you are done with your hay infusion. Hopefully you found some really awesome organisms. You can wash your hay, you can wash this jar, you are now done, or let your Yeti drink it. Oh gosh, let your Yeti drink it. The only other part to this lab is that you're actually gonna, it's called photojournal and organelle. So you don't need any of the, I'm gonna shut my computer because Yeti is a mess. You wanna find something in your life where you can find, something in your life, in your house or go outside, that looks kind of like the 3 organelles. 3 organelles that you choose. So like, for example, I think roses sometimes look a little bit like Golgi, the Golgi apparatus. So you're gonna find something in nature that looks kind of like an organelle. Take a picture of it or draw it, and then why do you think it looks that way? It doesn't have to be a picture, you can draw it too. And it doesn't have to be outside, it could be something inside your house. So I actually can see something right now. My kitchen is not that clean, I apologize. Oh gosh, Yeti. Yeti, actually, will you go get that plant right there, the second one in, that looks, it looks like cytoskeleton. So I just want to show you, I just saw something in my house that looks like an organelle, part of the cytoskeleton, and hopefully White Yeti won't break it. White Yeti should live outside. So, no, okay, there you go, good job. Okay, nice, White Yeti. This plant, to me, because of the branching, looks a little bit like microtubules, okay? So I could take a picture of this or draw it and say this plant, it looks like microtubules to me because it kind of looks like they're these long strands that are like highways around the cell. Does that make sense? You'll do this for 3 of them. And then you're done with your lab. Okay, White Yeti, can you say goodbye? Bye.